Hello, everyone, and welcome to phyloseminar.org. The current theme is transmission trees, and this is the first talk in a series of 3 talks on that topic. Please use the YouTube Live comment box to ask questions or tweet at phyloseminar. Today's speaker is Colijn. Colijn works at the interface of mathematics, evolution, infection, and public health. She did her PhD in applied mathematics at the University of Waterloo. Her first postdoc, which is when she started working on biology, was at McGill, and the second was at Harvard and the Broad Institute. She started her faculty career at the Department of Engineering Mathematics in Bristol, England at 2007, and then joined Imperial College London's Department of Mathematics in 2011. She moved to SFU's Mathematics Department in 2018 as a Canada 150 Research Chair in Mathematics for Infection, Evolution, and Public Health. Welcome, Colijn, and thanks for speaking to us today. And actually, maybe before we start, you should actually correct me on the pronunciation of your last name.
Yeah, sure, Eric. Thank you so much for having me. It's Colijn. Okay. And I'm really excited to be part of the Philo Seminar series. I think it's a great resource for the community, and you should all go and look at the other talks, which you can see, all the past talks up there. You can watch them on the internet, and it's a great resource. Okay, so I'm going to jump right in. I'm going to talk about genomic epidemiology with TransPhylo, methods, applications, and limitations. And I'll get back to this at the end, but I wanted to just make a few topical comments about the new coronavirus, which you've heard about. And I think there's some fantastic open data out there and some fantastic open analyses that are being published. More and more are coming out every day, so you can read about that. The method that I'm going to talk to you about today is probably not ideal for an early outbreak analysis like this one, where there are already thousands of cases, but we only have about 43 sequences. So the method I'm going to talk about is more for densely sampled and densely sequenced outbreaks of an infection, which could be a new infection. But of course, with a new, rapidly spreading and emerging viral infection like the new coronavirus, we typically don't have sequences for most of the cases. So, of course, these talks were planned before the new coronavirus outbreak had happened. And, this is kind of within phylogenetics. This topic is very topical and related to that because we're talking about spreading behavior of person-to-person spreading. But you can't sort of rule out the method I'm going to tell you about today and figure out transmission about the new coronavirus with this tool, unfortunately. Okay, so as you've heard probably, we have now a lot of data for pathogens, in particular for things like influenza, but also for outbreaks where we really want to understand person-to-person transmission. And that's largely driven by the rapid and exponential decrease in cost of sequencing technologies. This is an NIH figure that is often used. You may have seen it before. And it just shows, compared to Moore's law, the exponential decay of prices of computing. Sequencing technologies fell off a cliff and dropped by orders of magnitude in cost over the past 10 or 12 years. So what that means for infectious disease is lots of things, but in particular, we can start to sequence people's pathogens. For example, tuberculosis, which is the bug that I'm gonna have in my applications here. And we can start trying to use that information to understand who infected whom and to understand broader patterns of the transmission dynamics of bugs. And we can do this lots of times, thousands of isolates, even in a single study in some cases, and even with bacteria, in addition to viral pathogens. Okay, so we want to understand in epidemiology transmission. Who is transmitting? What are they transmitting? What variants? When are they transmitting? Where are they transmitting? And why? What are the determinants of transmission? And these things are core questions in infectious disease epidemiology. And the hope, I think, with sequencing technologies having decreased in price so much, was that we would be able to use that information to better understand transmission. And of course, we have traditional methods, which are great and which are labor intensive and produce a lot of rich data. And we're hoping that these genomic technologies can now complement those data, and sometimes maybe we might hope that they could replace costly and time-consuming kind of person-to-person interview investigations. So far, I think the jury's out, and that'll be pretty context dependent whether that works. But we want to understand these processes. And we have sequences now, as I've just said. And with our sequences, we have our cartoon scientist here who can make a phylogeny. And that's this tree that is sketched here. I can zoom in on it a little bit. There it is. When you can just see it has nodes and edges, and obviously that's a cartoon that I drew by hand. But in that type of tree, the tips of the tree correspond to taxa, organisms, which are the sequences of the pathogens. And the internal nodes are inferred common ancestors of groups of tips. So those internal nodes are not directly observed, and the tips are directly observed. All right, so we want to use these objects, and we want to try to understand transmission using these objects that tell us about relatedness and ancestry. We want to understand kind of relatedness and ancestry among the person-to-person transmission events. So that seems promising. Okay, so it kind of goes like this. This is a cartoon made by Jennifer Gardy, who is a co-author on this work, and who's now at the Gates Foundation. So you probably remember the old game of telephone when you were a kid, and people would whisper messages to each other, you know, I like peas, I heard you sneeze, and so on, and it would go through, and then at the end, it's funny. It's kind of like that. But we may, with respiratory contact, be transmitting pathogens. And as they get transmitted, they may acquire small amounts of genetic variation, which we can measure with our fancy new, cheaper sequencing technologies. So the aim is to get these sequences, which I've sketched as AECs and so on, and reconstruct our trees and figure out who infected whom. Okay, so far so good. So I want to describe another kind of tree, which is the tree we want to infer, namely a transmission tree. So it's a different object than a phylogeny, which I just described. So a transmission tree is a tree in which the nodes are people and the edges correspond to infection events. So in this one, case 60 infected case 68, who infected case 73. And you might annotate this object in lots of ways if you want. Colors could represent time, as it does here. Squares and circles could represent types of people, maybe sampled or unsampled. And edges, you may want to record or estimate the time of that event. So when did 60 infect 68? And with what confidence did 60 infect 68? So these are some of the things that we want to understand and that we want to draw on an object called a transmission tree. And I want to point out that transmission trees and phylogenetic trees are not the same thing, right? Here, the nodes, internal nodes, and the leaves are people, and they're corresponding to the sequences of their pathogens. And in the phylogenetic tree, the tips were sequences, and the internal nodes were these unobserved, estimated ancestors. And so these are kind of different objects. All right, here are two of them, and these are actually consistent, which I'll draw in a little while. So these are just two examples. Here's a transmission tree, case A infected B and C. And here's a phylogeny, A, B, and C are all tips. And there's some inferred ancestors among them describing the shared ancestry of the sequences. All right. So what we want to do is explore these relationships between phylogenetic trees and transmission trees. And I think when sequencing got so cheap, there was a lot of excitement around, okay, we can sequence and then we'll know who infected whom. We're just going to know it because we're going to make these trees. And there was kind of a confusion between phylogenetic trees and transmission trees and when they might be the same or different. But actually, it turns out you can't just read off who infected whom. So, for example, I made up all these names, by the way. This is a completely made up tree. And let's focus on this pair down here, Rohanna and Shannon. I think there was a kind of early assumption that transmission events would always show up as pairs in these trees. And that makes sense if sampling is really sparse and these two are super closely related, then they might be, in the context of, you're only sampling 1 in 500 cases and you really find this close event, that might make sense. But if you have dense sampling, think about it, we make a binary tree, a phylogeny. You can only be in a pair with one other person, but you could infect many of your unfortunate friends. And so, of course, not all transmission events can appear as pairs or cherries in a phylogeny. And equally, not all closely related pairs of sequences, for example, these two, necessarily have direct transmission events. So we can't really read off from this tree the story of who infected whom. We can make up some stories. Here's Ikra, she infected John, John infected Catherine, Catherine infected one of these two. We can read that off, and it might be right, but actually it might not be. Okay, so we found in many of our papers on this topic that, especially in an organism like tuberculosis, where relatively low amounts of genetic variation occur, and even in other organisms, having sequencing doesn't directly reveal who infected whom. And there are different sources of uncertainty and error that drive that lack of direct revelation. One is, that we don't always know how closely related transmission pairs might be, particularly in a chronic pathogen like TB. Latent and infectious periods can be very long and variable, so transmission pairs could be over widely ranging periods of time. People can harbor diverse infections. Times of diagnosis might not have a lot to do with times of the beginning of infectivity. Set cases can be unsampled. We don't always know all of them, and we don't know kind of what bacterial or viral particles are transmitted and how many of them at the point of transmission. So all of these things complicate the relationship between getting sequencing with our nice new cheaper sequencing technologies and reconstructing who infected whom. Okay, so that's our setup, and what we want to do is understand that relationship and make methods to infer who infected whom with uncertainty in this context of these complications. So I wanted to focus just for a minute on in-host diversity, and I've got my 3 cartoon people. Maybe they can be cartoon scientists in this case. And this one with the gray hair has quite a diverse infection, and he or she infects 2 others, this one and this one. And they, of course, start their own little viral or bacterial populations, which start to diversify. And then subsequent to that, they get sampled, they get ill, that's sad, and there's notified, and they get sequenced, their samples get sequenced. So here's the true phylogeny among these cases. We have this gray, and we've just imagined that we can draw the information all together, and then these 2 lineages went off and infected these unfortunate friends, and then later they get sampled. So the true phylogeny here among the stars, the sampled cases, would have these 2 being a pair, and this one being off to the side, just like my ABC example before. And the true transmission tree is that this case infected both of those. All right, so transmission pairs are not necessarily phylogenetic pairs. And this is in-host diversity is a way that can happen, or probably the only way that can happen. Okay, so how do we resolve that? Or how do we quantify and work quantitatively with those relationships? We do that through coloring. We're going to draw the trees and color them in. And then we're going to teach our computer how to draw the trees and color them in, of course, and do that in an automated way. So just for the sake of this conversation, I'm going to say a lineage means just section of a branch of a tree, some line in the tree. That's not what it means in some communities. Okay, and we're going to just draw some reasonable constraints for a person-to-person model. So a person can have more than one bacteria or lineage at a time. So person A, here's person A's diverse infection. This was the gray-headed one in the previous example. But each lineage, each line in the tree can only be in one person at a time. That's what the phylogeny means. And when it changes from person to person, that's a transmission event. So this pink lineage, this pink line can move into host B, and this color change here is a transmission event. And in this way, we can take our phylogeny, the black lines connecting A, B, and C here, and we can draw a story of who infected whom on top of that phylogeny. So okay, we can draw it. So what? I'll tell you so what in a minute. But in that way, we can explore what transmission stories are consistent with our genomic data through the lens of our phylogeny. Okay, so we call this thing an admissible coloring. Every host has to have a color and only one color. Not all hosts have to be sampled. Each lineage is in one host at each time. It only has one color, and the colors can't be broken up. They have to be continuous. So this green here is all, you can draw the whole green without lifting up your pen. All right, and that tells a story of person-to-person transmission in which each person only has one infection. If you want to add more infections per person, you have to do some tricks here. Okay, so that's the coloring. That's how we color them in. Here's an example of a valid coloring for a TB outbreak. This is in our 2014 paper. So here we have this 35 as the index case. They were infected first, and they had a lung and chronic infection. At this time, infected, this case, KO4, and so on. So you can read off the story of who infected whom and when by reading who the lineage is in. Here's the color and which case it is, and then when the case infected other people. So one of the things that is an advantage of this method is that we're able to respect the constraints that the genomic data give us on who might have infected whom. So for example, in this tree, so time is going down from top to bottom, and we have some constraints. So it could be the case that B infected A, right? So if B infected A, then we have to draw B's color all the way up from B to the top, and then somewhere along this line, the color would have to change to A's color. So if B infected A, then this is all B's color, and B would also have to have infected C, right? If B infected A, then B infected C. We wouldn't get that from a pairwise model that just looked at genomic distances between B and A and C and A. Similarly, if A infected B early, let's say A infected B up here, then B must have infected C, because this lineage would then be colored with B's color, and the only way for C to get it with these continuous color rules is for B to have infected C. And if C infected A, then C infected B. That's symmetric from the first statement, and so on. So even in this little toy example, you can see that the shape of making sure these genomes are in a tree will constrain the set of transmission events that are consistent with the genomic data. And that's where this method comes into play. So we're going to make this coloring, and then we use the coloring to find a likelihood. And we write down our likelihood in two major components. The first is the likelihood of the transmission events, that the color changes on the trees. And that is based on our epidemiological parameters that define our transmission process. And that comes from our knowledge of the pathogen, the sampling process, the natural history, the person-to-person contact information, and that kind of model. The other component of our likelihood is the phylogenetic component. And that is greatly simplified by the fact that we've drawn these colors on the tree, because those transmissions break our genealogy or phylogeny G into little independent trees, each in a different host. So each color corresponds to a little mini tree that is inside a different human being or a different host, if it's not humans. And then those trees are independent. So that likelihood for the genetic data part breaks up into a product of simple terms. So I won't talk as much about this phylogenetic component. I'm going to focus more on the transmission component, because that's where we made an innovation here.
So the branching process model, you were saying that that's pretty much like an epidemiological model. And are you going to get into that more?
Yep.
Okay.
That's going to be next.
And I guess you're not going to talk much about the phylogenetic likelihood, but it seems like the coalescent model does it make the most sense if you have multiple samples per individual, or is it even sort of useful to have even if you only have a single sample?
So it's okay if we have a single sample per individual, in the sense that we may have individuals with whose in-host phylogeny in our model has multiple tips. Some of those tips correspond to transmission events from that person to another person. Those are kind of augmented because we don't observe those, but they still play a part in the coalescent likelihood. So the coalescent model is for the within host trees. And we know which tree is within the host, or we know because we've augmented our tree and colored in these transmission events.
Thanks.
Yep. Okay. So you can read about this in the papers, of course. The decomposition given a fixed phylogenetic tree looks like this. So we have a likelihood of our transmission given our phylogeny. And we look at the likelihood of the transmission events, these color changes, the likelihood of the phylogeny given the transmission events, and then the priors. And it's this part that allows us to separate the one big phylogeny into lots of little phylogenies that are independent and use a within-host coalescent model for each one. The transmission events likelihood comes from an epidemic model for the system. And we take into account that the study ended at a finite time, and that's something that a lot of branching process models don't do, because most of the theory of branching processes comes from either finite time or letting time go off to infinity. Okay. So here's some information about the model within TransPhylo. We always model a negative binomial offspring distribution, and what that means in epidemiology land is that the distribution of the number of secondary cases is negative binomial, so it's over-dispersed. That in turn comes from a Poisson number with a gamma distributed rate, in case you're curious. And I want to just note that this distribution has a convenient generating function, which we use in a couple of slides. Okay, let's imagine that I'm infected at time t, little t, and the study ends at time capital T. If I'm infected today and the study ends tomorrow, the probability that I'm sampled is very low, and we take that into account. So we have an overall sampling probability, and then we have a time to sampling, which we describe with some density function f sub s. And if the time of infection, little t, is really close to big T, then this integral is small. And so we take into account that people sampled, people who were infected yesterday may not have been known in the process if the study ends today. And then after the study ends, no one got sampled. And we have to make this assumption for technical reasons right at exactly the time that I'm infected, I don't infect others right then. So FG is the density for the generation time, which is the time between someone getting infected and the time that they infect their secondary cases. So we have this finite time assumption, and we also model the fact that not all the cases are sampled. And that's something that a lot of the methods in genomic epi make an explicit or implicit assumption that we know about all the cases. But one of the mathematical challenges of this work was actually dealing with that challenge. So I'm going to take you through the transmission likelihood a little bit and how we managed, or how we solved this problem of unknown cases, the unknown unknowns in the model. Okay, so we're going to let P naught of T be the probability of being unsampled and having all your descendants unsampled, so all your secondary infections, all their secondary infections, and so on, everybody, forever unknown, if you were infected at time little t. Okay, so let's imagine we have our classic branching processes. T is going to go out to minus infinity, so this outbreak took place forever ago in the 1800s, and somehow we have sequences. Okay, so it took place forever ago. We expect this to converge to some number, fixed number, and it turns out that's kind of a standard thing in branching processes, this usual equation where in order for the person to be unsampled and all of their descendants unsampled, they themselves have to be unsampled, right? So if pi is the sampling probability, then 1 minus pi is that they themselves were unsampled. And no matter how many secondary cases they had, k, all of those also had to be unsampled and all of their descendants unsampled. Okay, so that's P naught of T, as T goes out to minus infinity, would have to satisfy this equation. So that's nice, and you can solve this with generating functions, and that's a very standard thing to do if you're into your branching processes. But in our case, we have a finite time T, and we know that if you are sampled at time T, capital T, when the study ends, you won't be sampled, and all of your descendants will also not be sampled because they have to come after you, and our study ended at time t. So here's a little bit of math. If you like math, this is for you. If you don't like math, you can just let it wash over you and get back to just sort of get the basic ideas. Okay, so our starting point is we want to estimate this same number, P naught, but now we don't want to have the star. We don't want to let the time go back to the 1800s. So we want to estimate the probability that someone is unsampled and they have no sampled descendants in the process if they're infected at time t. So like the other equation, they themselves have to be unsampled. And no matter how many offspring they had at times tj, j is 1 to k, those guys also had to be unsampled. So it's the same idea, but now we have time in it. Okay, so this is the probability that they themselves are unsampled given infection at fixed time t, which I just showed you from our sampling density little integral. And now we do this same conditioning. So this is notation that means this, the probability that they had k offspring given a starting time of little t. And then for each of those offspring, j is 1 to k, they all had to be unsampled. And that's a little hard to compute because now we don't just go out to t is minus infinity. We have to say, well, when did the case have those descendants? They had them at these unknown times, tau j. So we have to integrate out tau j. This is the probability they were all unsampled if that the case j was unsampled having been infected at time tau j. And this is the density that case, the j's case, was infected at time tau j based on our information about our generation time. So fg is our generation time density. And that might mean that if it's coronavirus and the incubation period is 4 to 10 days, then that affects the likelihood that someone infected their friend 4 days after they were infected. So tau j minus t would be 4 days. This thing would peak. If it's TB, this thing might be 6 months to 2 years, for example. So that's really our epidemiological model. Okay, so I've highlighted this integral in red and called it thing. And that's because we have this thing here in the brackets, and we have a product of k of them. So that's thing to the k, right? This integral term all to the k. And here we have p sub k, the probability that you have k descendants. And now we can exploit the fact that we know the distribution of the number of secondary cases, and it has a nice generating function. And we'll call that generating function f of s. This here is just a definition of a generating function. Put in a number s, and this object defines the generating function for a distribution. And these are known for lots of common distributions, all of the common distributions. So if p sub k is negative binomial, then this is the form of this p sub k of t. That shouldn't be 0, that should be a p sub k. Oh no, p naught of t, no, this is right. This is p naught of t because we have the 1 minus pi t here. And then this expression looks just like this one, except instead of s, it has thing in it, right? So there's thing in the generating function for the distribution for p sub k. All right, so what? Now I've put this thing here, raised it to the power k, and I've put it in the generating function for the negative binomial distribution. Who cares? Well, it turns out this we can actually solve. The thing has p naught in it, right? Because p naught was in that integral. But if we discretize that integral, we can actually just numerically compute it with the trapezoid method. And we can solve this equation for the unknown function p naught of t. Okay, so again, you're thinking, okay, you did a bit of math and you solved something numerically. So what? Well, that gives us the probability that someone is unsampled and has no sampled descendants, conditional on having been infected at time little t. Right? So that gives us information about how to write down the likelihood of the transmission tree that we have drawn in the model, because someone in our model might have infected k others in total, but only some of them got sampled. And for the rest, the k minus d naught of them who did not get sampled, we need this information, this p naught of t, this probability of all of those other ones, these unknown unknowns that they infected and then who never got sampled and who never got any descendants. So of all their K in total that they infected, only D naught of them were sampled. So we have K choose D naught ways to do that. The probability they infected K, the probability the remaining K minus D naught were unsampled and had no sampled descendants. And then the term for the remaining D naught had to be sampled. Okay, and we can compute this. Typically, D naught is small. We typically are working with outbreaks of up to a few hundred cases, and so we never have, one infecting 300 others, for example. All right, so that allows us to build up the transmission likelihood, and I've color-coded it for you because the equation looks a little bit messy, but because this is online, if you really want to know, you can, of course, pause when you're not streaming and take a look. So I've just put the notation here. 0 and 1 for SI encode whether host I was sampled or not. These times, infection and sampling, inf and S, are the times of infection and sampling. And then we have 3 major ideas in the likelihood. So the first idea is in red. Was the case sampled or not? The likelihood depends on the time of sampling, how fast are we finding cases, and on the time of infection. How long before the end of the study were they infected? If they were sampled, and if not, they just weren't sampled. Okay, so that's idea 1. Idea 2 is the one we just talked about, the blue idea here. That is the probability of the number of sampled descendants that they had, including this idea they might have some unsampled ones who were unknown and who never had any sampled descendants. So that's the blue we just computed with our trapezoid method. And then for the sampled descendants, so the descendants that we know about in our tree, we have the density for the generation time of when they were infected. So these 3 ideas build up our transmission likelihood. Timing when you infected your descending cases, how many descending cases you had, and the timing and nature of whether you were sampled for all you in the outbreak, so for all of the cases in the tree. And that gives us our transmission likelihood. All right.
Can I just butt in one more time?
Sure.
I'm curious. So, I mean, I understand that this is definitely doing a lot more than sort of the usual phylogenetic thing, and it's doing a nice job of bringing in some ideas about how the infection works. But there's definitely some limitations about the richness of the model that we can express using something like this. Like, we couldn't say, like, Frida is from one neighborhood and Joe is in the same neighborhood, and they sort of have they're more likely to infect each other. I mean, there's some exchangeability things and things like that. I mean, what are the assumptions? What is the sort of richest model that you can put into something like that?
Yeah, so I mean, I think you're right. When we do this, here, we are integrating out exactly one unknown quantity, which is the unknown time of infection of these secondary cases. We can't be integrating out, okay, now what neighborhood were they in? Okay, were they smear positive cases? Okay, what were their clinical symptoms? We can't just be taking all these unknown unknowns and putting them in the same framework. What we can do is put that information in the prior, and that's what we are doing. So if we have prior knowledge that these people were separated by a long distance and they likely didn't infect each other, we can put that knowledge on our transmission tree prior. And then we can look at our posterior, but we can't formally estimate parameters connected to covariates and how they affect transmissibility.
Should I have seen that prior expression before, or are we going to see it later?
We are going to see that later.
Perfect. All right. I'm always interrupting in the way. But
yeah, no, that's great. It's good because it confirms to me that you're still hearing me.
That's very helpful.
Yeah. Okay. Good. So what do we do with our likelihood? We have this big likelihood. Well, of course, what we do is we roll it out in a big MCMC Markov chain Monte Carlo inference. We start with, for now, one fixed phylogenetic tree in units of time. That was the black object with the lines and the tips. And we start with our information about our epidemiological model and some priors. And as our method goes through at each iteration, we propose a coloring, which is who infected whom and when. We compute its likelihood using our 3-idea model from the previous slide. We compute the likelihood for the independent mini trees that are inside each host. So conditional on those transmission events, we divide up our big tree into lots of little trees inside different people. We accept or reject our proposal, we continue, we propose a new coloring, and so on. And at the end, we have a giant collection of these augmented phylogenetic trees, which are colored in according to who infected whom and when. So altogether, it takes in a fixed phylogenetic tree and some priors and produces a bunch of colored objects. Who cares? Well, who cares? We hope that people care because this knowledge is useful. In particular, we can extract from our transmission trees who infected whom and when. For an individual, what's the posterior time of infection for that individual? Where were there unsampled cases? Posterior estimates of how long between infection and infecting others, between infection and sampling, and where there might have been missing cases, which could mean that we even direct contact tracing to say, hey, we don't have a credible infector for case G92, but we do have one for case G17. So we hope that that will be useful, and indeed, I think this is being used in public health in a number of different settings. Okay, so what data does the method need? If you are a person out there and you want to use this method, what data do you need? Somewhat unfortunately, you need a timed phylogenetic tree or a collection of them. You need sampling dates for the tips, which you need to make the timed phylogenetic tree. You need prior information about the time between getting infected and infecting someone else. It doesn't have to be a sharp prior, but you need some prior information. Also, you need information about the time between getting infected and getting sampled, overall probability of being sampled, and some information about the time when sampling stopped. And that's the minimal. The more priors you can add in, of course, the more refined your posterior becomes. That's the nature of Bayesian thinking. And what does the method produce? So formally, it estimates 3 key parameters: the mean of the offspring distribution, or R naught, the in-host effective population size, and the sampling fraction. But in practice, that's not, I wouldn't necessarily use this method as an R naught estimation method because it's a tree. Everyone has one infector, and then these unsampled cases. In practice, I would use this as a posterior collection of who infected whom stories that are consistent with your genetic data, posterior generation times and times between infection and sampling, and posterior estimates of the locations and numbers of unsampled cases. Okay, so that's what it produces, and you can pull out all manner of colored trees from this method. All right, so I'll give a couple of applications. I called the talk the method, the applications, and limitations. So now I've told you how the method works, and I'll tell you about some applications. So in this paper in Microbial Genomics, we analyzed a set of TB cases from Norway, from a strain that's associated with people coming into Norway from countries in the Horn of Africa, primarily, and also elsewhere. And often in Norway, since there isn't a lot of TB in Norway, often people who have TB, their country of origin is somewhere else, and that place has a higher TB burden than in Norway. And so there's kind of a natural assumption maybe that could be made that, oh, well, this person came from a country in sub-Saharan Africa where there's lots of TB. They must have had their TB exposure there and then reactivated when they moved to Norway or sometime after they moved to Norway. But my colleague Vegard Eldholm saw in the genomics that there were some closely related cases that were suggestive of recent transmission. And if the transmission is recent, then it might be happening in Norway. And that might trigger different public health interventions than if the assumption is that people are bringing their TB from elsewhere into Norway and reactivating after that. But of course, with TB, we really don't know a lot about the generation time. It's not a flu-like illness. It's a chronic infection. It could be months or years between infecting and infecting others. It has a long and variable latent period. And we don't know how rapidly we would be finding cases. So we can put priors in this model to reflect that uncertainty. And we just wanted to ask the question, is there likely to have been transmission within Norway? And the answer is yes for some of the cases, unknown for some of the cases, and no for some of the cases. But we can do an individual case-by-case posterior to look at that. So here, each panel is a case. The red blobs are the posterior density of when the case was infected in our model. And the blue information is when the case moved to Norway. And for some individuals, they knew exactly when they moved, here 2001. And for other individuals, they only had a range. They weren't sure exactly when the person arrived in Norway. So if you just look at the amount of red that is after the blue, you can see that in some cases we have a very strong indication that the person was infected after they moved, even though we don't know when they were infected very sharply, but they moved so long ago that it's very likely to have been after they moved. In other cases, in this case, for example, there's probably about a 50-50 chance that they were infected after they moved. Maybe they moved at the middle of this time, and if you look at the mass above that and integrate over this box, then you might get 50-50. And for others, it's very clear that in our posterior that they were infected sometime in the few years before the data were sampled. Yeah, so there is some fairly strong evidence here that there is some transmission happening in Norway, and therefore that we shouldn't just always assume that if people have a country of origin where there's TB, then that must have been where they had their TB. And that's published in Microbial Genomics 2018. I should say the method, I didn't put a link to the paper for the TransPhylo method. There are 2 papers about it, both in molecular biology and evolution, co-authored with me, Xavier Didolo, Christophe Fraser, and Jen Gardy. So you can look for those. Okay, so here's another application, and we've made a few extensions of the method in the past few years, and I want to highlight some of those, at least one of those. So, of course, if you're working in infectious disease, you may have the following happen, where you don't have one big tree that represents a lot of closely related, densely sampled and densely sequenced cases. You may have lots of little clusters. And if you run, so here's a cartoon of 3 very well separated little clusters. You have a cluster from some TB lineage and a different one and a different one. Maybe they're separated by hundreds of polymorphisms or many years. If you just throw this into TransPhylo and you run it, what it's going to do is it's going to spend a ton of computational effort placing unsampled cases on these really long branches. And that doesn't give you any information because you haven't sampled cases along these long branches. And what it won't be able to do is explore transmissions within the clusters very efficiently. And that's what you'd be interested in. So Yang-Wei Zhu, who is a postdoc in London, and I developed a method to share parameters between lots of little trees. Instead of putting one big tree into the method, we put lots of little trees into the method. And that can happen, as on the previous slide, because you have different clusters. Or it could happen if you want to simultaneously analyze many input phylogenetic trees, because you don't believe you have one true tree, you have an uncertain tree, and you want to sort of run over all of them, but you want to make sure that they share the same proposals for the 3 key estimated parameters. So Yang-Wei introduced this parameter sharing. So formally, as I said, the method can estimate these 3 things. If you have n clusters, you would be estimating 3n parameters if you just ran the method separately on the n clusters. If we share the parameters, then we can estimate 3 instead of 3n, so that's a nice savings. And it also means that what is a poor likelihood in one cluster can actually affect the acceptance probability and affect the rest of the inference. So the clusters can affect each other, which wouldn't happen if we just ran them all separately and got 3n separate estimates. Okay. So formally, you can see this way. If you have n trees, each one corresponding to a cluster or each one corresponding to a posterior sample of a phylogeny, if you don't share parameters and you need to estimate theta, your parameters for each tree, then you have n of these. And if you can share them, then you only estimate one set of parameters for all the trees. And this is in the main TransPhylo GitHub, which is on Xavier Didolo's GitHub. So you can use this and you can share some or all of the parameters across the trees. All right, so here's an application of that from Valencia. This is data provided by Iñaki Comas and his group, and it's published in PLOS Medicine. We had 110 transmission clusters of TB in Valencia. Many of them were very small, so 1 or 2 cases, and then we don't run our TransPhylo method. But for bigger clusters, we can run it. I think we had 24 clusters that were big enough. I think 5 or 6 cases and up. And this is not my picture. This is Iñaki Comash's picture, just illustrating the clusters in this color-coded way. And you can sort of see their size. So why did we do this? Well, Iñaki was interested in looking at whether the first diagnosed case was actually the index case of the clusters. And that's something we don't know the true index case, but with TransPhylo, we can run our method and see whether we have strong posterior support for a particular index case or not. We were also interested in whether there was evidence of transmission before the reports of symptoms. So people may be sort of coughing, but sort of not too severely, and was there evidence of that? How long between infection and infecting, infection and sampling. Iñaki was also interested in, whether this is really, as public health people there tended to interpret, A infects B, infects C, infects D, infects E in a chain, or were there more complex patterns? And are there clusters where we're still missing the index case and where it might be worth doing a bit more investigation to go and find potentially circulating undetected cases to stop transmission? Okay, so we need our timed trees, and that's a little dodgy, but we did it anyway. We used tree data to date the phylogenies, and we used a range of different clock rates to explore uncertainty to the clock, because we can't estimate the clock with these data. So here's a smattering of a few of our clusters in the timed trees. And we ran our method over lots of clock rates and lots of iterations, and we found that there were 4 of the clusters of these 10 shown here where there was evidence at reasonable clock rates for TB that there were more unsampled cases than others. So we sort of had 2 groups of clusters. These ones are where you would want to look if you wanted to go and find unsampled cases. These ones, not much evidence of unsampled cases. We found, as you might expect, that transmission events are still quite uncertain. This is a schematic kind of network picture of one of the clusters. The circles are cases. This one is unsampled, and this one is the unknown source elsewhere in the world for the cluster. The edges reflect transmission events that came up in our posterior with sufficient probability to be in the picture, and the edge thickness reflects the posterior probability. So this is an event we're pretty sure about. This is an event we're pretty sure about, so that would make this one the index case for the cluster, or this unsampled index case. This one was the first one in the cluster that we got, and then these other events we're not so sure about. But one of the things that comes out of this analysis is, for example, the transmission events coming out of this node have very thin edges, right? G1761, not a lot of posterior probability that this one infected anyone else. Whereas this one, we have a pretty strong edge, 2 strong edges coming out. So we can separate, who is a likely transmitter or a credible transmitter in the model from other clustered cases. And that's important because we can then start doing analysis downstream of that, of what properties did the credible transmitters have, as opposed to just any case that was in a cluster. Here are some of the results for the question, was the first case who was diagnosed in a cluster the index case? These numbers psamp at the top indicate the probability that the index case was sampled. So of our posterior, how many of our posterior trees had a sampled index case for the cluster, and then when that sampled index case was. So in this cluster, the index case, the most credible index case was also the first diagnosed case. But in this cluster, there's strong indication, at least by our model, that the first case who was diagnosed was not the index case. And that's something that they were interested in finding out for these clusters. Okay, so I'm going to talk about one more extension by a student called Matthew Gould here at SFU, and here's his GitHub. And this gets back to Eric's question. So what if we know that we have 2 neighborhoods or 3 neighborhoods, and these 2 cases are just not in the same neighborhood? We can't build that into our likelihood, as we discussed with the branching process, but we can build it into the prior. So we can just say, okay, transmission tree, if you break my prior knowledge, you get a penalty by having a lower prior. And that, of course, will shift our posterior towards the knowledge that we have. So right now, Matthew has implemented locations. So you can penalize transmissions whose individuals were in very different locations, and you know that. You can also restrict or penalize transmissions that break plausible transmission times. So if someone didn't move into the community before a certain date and wasn't there, they can't have been transmitting. If they had a negative screen at a certain date, they shouldn't be transmitting before that date. If they left the community or if they died, they shouldn't be transmitting after that date, and so on. We can build in times for symptoms, and we can also build in contact data. So if you want to run this method and you want to really make sure that your posteriors like really respect the knowledge that you already have, but add something with genomics to that, then this is for you. And you can go to Matthew's GitHub. He's got a demo on there and a simulation where you can simulate some data and make sure that your trees are kind of matching up to your epi data. All right, so heading towards the discussion. Before I get into all the limitations, some of which you've probably thought of during the talk, I want to just highlight a few of the advantages. So it includes genetic data and trees, phylogenetic trees, and unsampled cases. The augmentation approach has some, the transmission trees are consistent with the phylogeny, so they respect those constraints, the ones about A, B, and C, and if A infected B, then B had to infect C. And that gives them a much more constrained transmission space than we would have if we didn't look at the phylogeny. We have this posterior collection of augmented trees, these colored in objects where we can pull out who infected whom and when with uncertainty. So those are both advantages in terms of interpretation of the method and how much we might be able to get out, but also while still respecting that we have considerable uncertainty left over, even if we're using genomic data. We can point out where there are unsampled infectors and infectees and for whom we don't have a credible infector. We can distinguish credible infectors from all clustered cases and do downstream analyses. And we've done one of those using a couple of machine learning tools to associate covariate data to transmission status and so on. And we can scale this to large-ish data sets using this multiple tree simultaneously approach. So if you have 100 clusters and you want to analyze them together and have them inform each other, we can do that. If you have 10,000 cases, then, well, you probably don't really have one true tree and you might want to split them up. This is probably not going to work with 10,000 cases. Okay, so limitations. There are some really important limitations here. We model person-to-person transmission with dense sampling. We don't have an environmental reservoir. We don't have the sinks, the taps, the kitchens, the ponds, the mosquitoes, the parasites, the food, the point source, the farm, the lettuce. So this is really a person-to-person model. And it really aims to model direct transmission events. So if you have only 5% sampling or less than about 75% sampling even, you may want to use another method because modeling direct transmission events, if you only have a third of the cases, you will have so few transmission events that you're better off with a pairwise model because this model will just put unsampled cases everywhere and waste a bunch of your time. Currently, it models a fixed sampling probability over time, and then this thing about the time before the study ends, that we could change, we just haven't done it yet. I think a bigger limitation is this two-stage aspect. So first, you want to make a timed phylogenetic tree, and then you model transmission on that tree. If that tree is noisy or you don't like it or you don't have one true tree, then the advantage of this method is somewhat lost. And we cope with that by putting in lots of Bayesian trees, but that slows the method down. So it might be good to develop methods that simultaneously reconstruct the phylogenetic tree and the transmission tree. These exist. One is Beastlier by Matthew Hall, who's on phyloseminar already, and one is Scotty, which models more of an environmental reservoir than unsampled actual individual humans. Beastlier does not have the unsampled cases. So Beastlier does this simultaneous reconstruction, but it doesn't allow you to not know about all the people in your outbreak. And in my view, that's a bit restrictive. Okay, and then this is the limitation that Eric saw right away. We have this branching process model, and we integrate out unsampled cases, unknown times of infection. But we can't integrate out over their unknown values of location, demographics, clinical history, prison history, smear positivity, symptom strength, symptom time, and so on. So the best we can do is use priors to incorporate that information if we want. And of course, we can run our model and look at the posterior and see what it says, if anything, about these covariates. But if what you want to do is estimate the effective prison history formally as a parameter, then this model is not going to be able to do that. I suspect that a survival analysis likelihood might be able to do that, and I've had some chats with Evan Kenna about it. He's upcoming in this same series, so watch his talk too, because he uses survival analysis tools to write down likelihoods around transmission processes and outbreaks. All right, I want to thank my group here, Matthew Gould, who did the epi prior coding, and Jessica Stockdale, who's been working on this with me as well, and people who worked on the results I've shown you, in particular Yanwei Zhu, who worked with the Valencia data. I should have said Priya Ayabina, my PhD student at Imperial, who's now, I think, at Warwick, Vegard Eldholm from Norway, and the co-authors on the TransPhylo papers. So thank you very much.
Thank you, Carolyn. So we'll take any other questions now that people have. I guess something you mentioned early on, and then I guess you had the TB example, was bacteria. Like, practically speaking, how should we go about applying this to bacteria? I guess you don't have to worry so much because you're sort of layering on this transmission likelihood, and that sort of doesn't really matter if it's viruses or bacteria. But I don't know, bacterial phylogeny seems kind of hard, or am I thinking wrong?
Right. I mean, I think that on the time scale of really closely related outbreaks, there are bacteria, many bacteria can actually be very clonal. We even have an outbreak of E. coli ST405 that we're looking at, which doesn't always look very clonal, but this particular one does. I think Staph aureus trees and strep pneumo trees from, on the very short time scales of person-to-person outbreak. Yes, these bugs recombine, but they don't recombine so often that you're likely to see it in the 1 year time frame of your outbreak in your closely related isolates. So you could still use a tree. If you start looking at Pseudomonas and other like really sink bugs, I mean, this is just not going to be the model for you, not just because of the phylogeny, but because of the person-to-person restriction and this coloring in method. One of the things I think would be fun to do is make up new methods for coloring in phylogenies that are not person-to-person transmission, and also to look at methods that don't require the 1 true tree or the 50 true trees. But I think my main answer to your question is on the relatively short time scales of person-to-person transmission. Even a bug like strep pneumo is going to not. There's going to be a core genome, and you're going to be able to make core phylogenies.
And that's a totally suitable answer. Thank you. So, I mean, you already mentioned this as being sort of a difficult part, which is the timing analysis. I mean, can you give some practical guidance about how people? I mean, if they just plop down a whole bunch of genomes. Would you suggest you've run some beast analyses, and you also had another one that I didn't know something about. It was
so tree data is Eric Voltz's R package. So there's basically 2 approaches to this. One is you can run beast. Typically in a person-to-person outbreak, you won't maybe want to estimate the clock rate from the data, but you will have a known clock rate from other estimates for your favorite bug. Or if you have 20 years of data, as we do in the Public Health England one that we're working on, then of course you might actually have enough to estimate a clock rate. But anyway, you either estimate or assume a clock rate, run beast, you get either a beautiful MCC tree with great posterior support, in which case, like, go ahead and run TransPhylo, because that's probably great. or maybe you get a sample of posterior trees from that. If you don't want to do that because you don't want to go through Bayesian phylogenetic reconstruction, you can make a maximum likelihood tree or a set of bootstrap trees, and then you can run them through a timing software that makes them into timed trees, and those include tree dater and least squares dating, LSD, and tree time, I think, also does this.
Okay. Cool.
Okay, do we have questions from the world, and if so, how do I see them?
We don't actually have any questions from the world, so world, ask questions. Can you just survey? I mean, as the co-organizer of this, can you just give us a little preview of like how Dawn? You've already mentioned Eben's talk, but how is Dawn's talk going to fit in?
Yeah, so Dawn is doing some really cool stuff. He actually has worked a lot on this limitation around simultaneous inference. So his models take the view that we don't start with a fixed phylogenetic tree, we simultaneously reconstruct the phylogeny and the transmission tree all in R, so you don't need to go through BEAST or tree dater or LSD or XML or anything like that. You can start with your genomes and your times and your information, and it will fibrate. Fibrate is the first one that he's done. That will simultaneously construct the trees for you within R. And then he's done some really interesting work on the bottleneck assumption, which I didn't talk about too much, but this model assumes that only exactly one lineage can be transmitted from one host to another host. And then it's immediately in this constant size population, constant effective population size coalescent model for within the hosts. What happens if multiple lineages and complex infections can be transmitted at the same time from person to person? Then this model is completely like not taking that into account at all. So he's done some really interesting work on that. I'm not sure exactly what parts of that he'll talk about. I would say the major methods that use phylogenetics in this field are Scotty and Beastlier, which you've already had in phyloseminar by Nicola de Maio and Matthew Hall, respectively. And then there's TransPhylo and Phybreak and Eben Kenna's survival analysis work, I think are, unless I've really missed something, I think those are the main ones that use trees. There are a lot of pairwise methods as well that look at genetic data from the point of view of the distances between pairs and then do Bayesian inference of transmission based on that. So they're using the genetic data, but they're not using this the full phylogeny. So for phyloseminar, I think we had suggested to span the complete space of genomic epi with phylogeny methods, which led to the choice of people here.
Cool. Well, we are out of time. We did have, I guess, a comment from Peter Van Heusden. It says, Iñaki Comas et al. Were doing the quote-unquote straightforward bacteria, i.e. M. tuberculosis.
Yeah. Yep, so TB is really clonal. It doesn't recombine at all. It's super clonal. If you can't make good trees for TB, from the point of view of recombination, you have a problem. TB, but I would stand by estimates of observable recombination or horizontal gene transfer rates in pneumo, Streptococcus pneumoniae, look like sort of one event per 10 years on a lineage going back through time in a tree from a core phylogeny. I think within person-to-person outbreaks, we can use these kinds of tree-based methods even for recombinogenic bacteria, because in a 3-month to 6-month time frame, most of the replication events are still descent events, and most of the genome is inherited from the one clonal parent, even in other bacteria. But yes, TB is one of our motivating bugs, because I have a background in TB, and because we can't. Another, I guess, point is that with these long and variable chronic infections, we can't use the timing to really inform who infected whom. So if I turn up today with flu or the novel coronavirus, I didn't get infected a year ago. With TB, you don't know that. So back to our coronavirus, you probably see now that if we have 43 sequences, which were how many were out there yesterday when I checked, but thousands of cases, this sort of really person-to-person, let's color in the tree is really not the right model for understanding data sets like that.
One more question, and maybe we'll close. Peter also asked, given a larger outbreak, e.g. some health districts in South Africa, with about 2,000 cases per year, would you be able to estimate approximately what proportion of cases were being detected?
Possibly. It's hard to say. But I would imagine that, what you'd want to do is, if you had, let's say 2,000, now imagine you had 2,000 sequences per year, that's a lot of sequences. You would hope to divide that up into clusters, each of which had some time in it. So over the past 5 years, there might be a cluster that was going on and had maybe 80 cases. And then you would have lots of those, and you could look at variable sampling rates or ascertainment rates between those clusters. But again, I mean, there might be multiple ways to get the same kinds of data. So it would be good to have priors on that that you would know from surveillance and from other data sources. But tentatively, yes, certainly varying, but relative rates between clusters, I think for sure we could do that.
Cool. All right. Well, thank you very much, and thanks for sort of doing a great job of really launching us off on this topic. And I guess, yes, next time we'll have Dawn on February 26th.
Great. I'm looking forward to it. Thank you so much.
All right. Thank you, Carolyn.
All right.